Good morning, good afternoon, and good evening to all of you attending today's webinar. We are excited to have all of you here on a very interesting topic, specifically about drugs of abuse in hair testing. And so first off, I would like to introduce myself. My name is Rachel Lieberman. I am currently the global market manager for forensics and toxicology here at Waters Corporation in our clinical business unit. And before we get started, I want to just give a brief update about what it is that you guys are looking at on your screen, kind of familiarize yourself with the On24 platform if you've never used it before. And I will be moderating for you in the background today and helping out with the question and answer session at the end. So here is a view of what the platform will look like. And what you will see is a question and answer box in the right-hand side. And we'd like for you to use that to ask any questions throughout the webinar. We will get to them at the end because we should have some time to answer them. And before we continue on, also in the center, you will see the slides for the presentation as well as the videos of myself and Jonathan. And then below the slides, you will find a resource list with any additional information and applications related to this particular topic. We will be having an application note on this data coming very soon. It's just not completed yet, but it's in the process of being finalized. On the right-hand side, you will find the speaker bio and a survey. Please feel free to fill out the survey at any point during the presentation. We really appreciate the feedback that you guys provide, as it helps us to make sure that we're bringing appropriate content to you guys moving forward. And if there's any topics that you guys want to hear about or see, please include them there. Additionally, we will be conducting a couple of polls throughout the presentation. So be prepared for those to ask some live questions and gauge the audience a little bit better while we're going through this. Lastly, at the bottom, you will see a little like scroll bar that will bring up any of these particular icons if you don't happen to see them on your screen. And there's also now on the right-hand side a little medal, and you can request a certificate of attendance if you need one for your continuing education credits. And you can just click on that, and then we will be able to provide that to you after this has completed. So without further ado, I would like to introduce our speaker for today. So today, our speaker is Dr. Jonathan Danisau. He is currently a principal scientist at Waters Corporation in our R&D division based out of Milford, Massachusetts. He received his bachelor's in biology from Allegheny College and a PhD in neuroscience from University of Utah. He joined Waters back in 2011 after spending several years in forensic toxicology and sports doping fields. And since he's been here at Waters, he's focused heavily on sample preparation and LC-MS/MS techniques for the forensic clinical research and other bioanalytical applications. So we consider him an expert in sample preparation and bioanalytical method development, as he's published many papers in this. And so we're really excited to hear him talk about this more recent work that he has done on drugs of abuse in hair. So his title is Extraction and Analysis of Comprehensive Quantitative Drug Panel in Hair Samples by UPLC, MS/MS, and Forensic Toxicology. All right, over to you, John.
Thank you, Rachel. And thank you everyone for attending today. I'm glad to see so many people here. I even see a couple of familiar names. Maybe I'll call you out later. And thanks for reading out that title so I don't have to one more time. And it's a good reminder. So this is a method that I've developed over the past several months and designed to extract a wide variety of drugs of abuse and commonly looked at, forensic compounds from hair. That includes opiates, amine stimulants, benzodiazepines, as well as, the standard drugs of abuse, such as PCP, cocaine. It does not have the cannabinoids. It has most of the other common ones. I did want to say that because I realized this morning I didn't have a list of them until 2/3 of the way through. So now at least we're all oriented. So this is the agenda I'll be talking about today. We'll first start with a hair testing background. Why do people want to test hair, and where and when do they need to test it? What are some of the advantages and disadvantages? It's getting more popular, but it's not a panacea like anything. And then we'll talk about the guidelines. What are the sensitivities we need to meet for which compounds? There are a lot of practical considerations that are very different from typical bioanalysis. For example, using bodily fluids like urine or blood or plasma, because it is a solid matrix. And we have to be concerned with things like contamination. So we need to do a decontamination step. We need to, because it's a solid matrix, we can't just dissolve drugs in it. We need to cut it or grind it or digest it some way to get at the cortex, the interior of the hair, to extract the drugs. There's often an incubation and extraction phase. That could be soaking in a solvent mixture. That could be, again, a digestion, something to break it down, etc. In our case, we use solid phase extraction to clean it up afterwards. However, many people, you can use liquid extraction, SLE techniques. For some people will simply dilute and shoot it. But I consider it fairly dirty for that sort of analysis. After that, we'll talk about the results, qualitative and quantitative. We'll look at the chromatography. We'll talk a little bit about the MS-Quant software that we use to analyze the data. We'll evaluate our extraction, looking at recovery and matrix effects and sort of the qualitative aspects of that. And then we'll look at our quantitative validation data and wrap it all up. So why use hair in testing teams? Well, many of you know this. There are a number of good reasons. Very non-invasive, convenient collection. You don't need any special training or equipment, just a pair of scissors and a way to hold it together. It's difficult to adulterate, unlike, for example, urine, where you can throw a pill in there or put a little bleach or something to adulterate it. Hair is much more difficult than that. It's very easy to transport and store. We don't have to worry, as in blood or plasma, of keeping things refrigerated to prevent their degradation. So those are very nice, some very nice features. It also has a long detection window, which is recent. It goes from the recent past to a fairly full history, or weeks and years of exposure to drugs because of the growth rate. So it's useful for long-term monitoring, things like that. It is often, I would say, complementary to other types of testing. It's not the end-all be-all. For many, for many situations, you can get much more accurate, metabolite of pharmaceutical or pharmacokinetic data from body fluid testing. But for all these reasons above, hair has become very popular and has a lot of advantages. So where is this used? As many of you know, drug-facilitated crimes, cause of death, any other situations where you may not have access to, urine or plasma, or it may be too late to get a good sample, or a sample where there's still drug in there. It can be used in workplace testing, long-term monitoring of truck drivers or bus drivers, that sort of thing. Sports doping. One of the challenges they have is, sample transport, and storage, especially for things like blood samples. So just like forensics, it can be, the collection makes it advantageous. And then finally, among other things, it can be used to monitor abstinence and detoxification programs. Again, the collection is easy, less privacy issues with urine collection, no needle sticks, and it can be a convenient way to do that. So these are sort of all the whys and the wheres of using hair testing. And as you can see, one of the nice things is it grows approximately 1 centimeter a month. So with a 12 centimeter hair, you can provide about 1 year of history, assuming they have long enough hair. So there's a number of factors that affect drug incorporation into hair, as well as its removal or degradation. And this is a little bit backwards, but we'll go with it anyways. One, you have incorporation from the blood. The blood vessels, feed and nourish the growing hair at the follicle, and you can get diffusion from the blood into the hair shaft itself. That, of course, can be affected by things like melanin content, and we'll talk about that in a little bit. You can get or deposition or incorporation from sebum or sweat on the surface of the hair shaft as it's, as you perspire, as the sebum is secreted to lubricate the hair shaft. So that's another way to get it. And then finally, you can have external or environmental causes. These are often things we want to control for to make sure we're not looking at incidental contact, such as dust, smoke, dirty hands, any sort of contamination that can get on the surface of the hair. And I mentioned before melanin content. Melanin is negatively charged, so it tends to attract basic compounds, like many of the drugs of abuse, amines, opiates, etc. However, it tends to exclude acidic compounds. This can make it a bit of a challenge for things like, the cannabinoids or ETG or ETS. So again, another possibly confounding factor that you don't have to deal with in other types of analysis. So how does it leave the hair, or how does it get removed or degrades? A number of things, fairly straightforward. Obviously, you can bleach it. That'll bleach degrades of lots of things, including drugs. UV radiation, somebody who spends a lot of time in the sun, shampoo, hair cosmetics, et cetera, those things can also help remove some of the drugs from hair. So we talked about alternative, other methods, and here's just a quick table of some of the advantages and disadvantages. I'm not going to go through all of it. It's fairly straightforward. Oops, I missed that one. One of the things with hair is often we don't have a whole lot of sample available. That was supposed to be highlighted, fell off. So often we're looking at, maybe 20 milligrams of hair versus, many mils of urine or a couple mils of plasma. So what are the guidelines and cutoffs? These are the guidelines from the Society of Hair Testing, as well as the European Workplace Drug Testing Society. And they pretty much, concur on most of, if not all of the compounds. The European guidelines and also include, alcohol, ETG, and a couple others. But as you can see, many of them are, in the sub nanogram per milligram range. From the 0.2 for the amphetamines down to, buprenorphine is at 0.01. So we need a fairly sensitive instrument system to make sure we can see these levels. So here's our first poll question here. Hopefully you guys can see it, but I cannot see the poll question. Rachel, do you know if they can see them?
Yes, they should be able to see the poll question.
Okay. So let's see. So I will give you guys some time to fill that out. Why don't we should we go ahead and move along and take a back look at that later, do you think?
That's up to you, but sure. If you want to go back, but yeah, so there you go. Now we have some answers.
Okay, gotcha.
Can you see the answers?
I can see the answers, yeah.
Okay, perfect.
They're starting to come in. I need to just refresh it.
So there's a good portion that are still not started or working on it. It looks like you got your answers right there.
Okay. All right, good. So a lot of people haven't started and some are working their way through it. So good to know we got a good spread. So let's look at a summary of some of the analytical challenges and requirements when we're analyzing hair. As I mentioned before, we're measuring from a single sample of hair, usually 20 milligrams or less. We need the sensitivity to meet the requirements of Society of Hair Testing and the EWDTS. We'd like this to be fairly high throughput. Again, it's not going to be as easy as urine or plasma, but we were looking to develop a method with a fairly short incubation time, and obviously, we need enough to make sure we get everything extracted, but in a reasonable amount of time, and a fast analysis as well, so we can look at the data quickly. We need a fairly simple and reliable sample preparation. Hair is a solid, complex matrix. It's very, very dirty. We see a lot of matrix effects, a lot of interference compared to other sorts of matrices. We're also looking at multiple analyte classes, so we need to compromise a little bit or sort of optimize it for the best possible results for everything. But we also wanted a method that was not too complex, that could be fairly easy to implement, straightforward, didn't require, very, tight pH windows or very particular steps in it. And then finally, a straightforward instrument system. In our case, we use our Acuity iClass Plus with Xevo TQ Absolute. It's our latest and greatest tandem quad, the most sensitive tandem instrument that we make. So these are all the things that we were trying to accomplish here. And now let's talk a little bit about the practical aspects of hair preparation. And this is where we'll get into some of the method development and the techniques that we use. So one of the first things most people, we need to do is to wash the hair. We want to remove surface contaminants that could confound results. We're looking for sort of ingestion or, systemic exposure versus environmental exposure. So if you look at the literature, almost all papers have a, wash with, a buffer or solvents or some combination of those. The next thing is we need to sort of pulverize, crush, cut, digest, whatever, but increase the surface area and really gain access to the cortex of the hair. And to the right, we see a picture from a journal article from a couple of years ago where they compared a number of pulverization or cutting techniques, and their conclusion was basically the more, fairly straightforward. The more pulverization, the greater surface area you have, the more efficient extraction they would get. So, like I said, many people will cut it up, some will pulverize it. We pulverize it with a tissue homogenizer. But however you do, you want to make sure that you are extracting what's inside the hair, not just what's on the surface of it. The next step is usually an incubation, often fairly high temperature and appropriate solvent system to extract the analytes. We're looking at a broad panel, so we were spending a lot of time optimizing that, making sure everything was extracted as well as we could and efficiently extracted as well. We need to make sure the pH is controlled as well. Some of the analytes are unstable under acidic conditions, some under extreme alkaline conditions, so fairly neutral or moderate pH, etc. Oftentimes, another technique is to digest it. We did not just digest our samples. We took a look at it, and I'll tell you later why we did not choose to do that. But that's another way to, certainly for certain more limited panels, it's a very good way to, really get access to all the air that basically dissolves it. And then finally, there can be usually an extraction or cleanup. We want to use, in our case, an appropriate SPE sorbent. You could use liquid, as I mentioned, or other techniques, but we do a cleanup in the end. So all of these are things, the washing, the pulverization, the incubation, and then the cleanup are things that need to be considered. So now that we've talked about this, let's look at our sample preparation workflow. What did we do? Well, we started preparing and incubate it. We decontaminated it with a series of washes, followed by pulverization. The hair was then weighed into a glass centrifuge tube. If we were running a curve, we would obviously spike our calibrators and QCs, internal standards for the other samples. Whoops, going the wrong way. One, yeah. And then we incubate it. We did a 2-hour incubation at 95 degrees with 1.2 mil of a developed incubation extraction buffer. That was followed by analyte extraction using Oasis MCX solid phase extraction plates. These are mixed mode cation exchange, and it's a method it's a extraction method we've used for similar panels in the past. And then finally, UPLC MSMS analysis on the TQ absolute. So how we prepared the hair is we collected from volunteers in the lab. We had a decontamination protocol, 6 sets of fritzes with an aqueous buffer as well as different organic solvents. We minced it in small segments, and then we pulverized it on the Prucellus Evo with their 2 mil lysine kit. 6 reps at 40 seconds, 6,200 RPM. And in the picture down below, or to the right, you can see this is a 2 mil tube with a screw cap, and you may be able to see spilling out there some ceramic beads of a couple of different sizes, and those do a very good job of just pulverizing the hair to a near powder. After that, we either extracted immediately or we stored it in room temperature in the dark. So here is our extraction technique, our pre-treatment and incubation. As I mentioned, 20 milligrams hair we put in the glass centrifuge tube. We added our internal standards, cals and QCs as necessary. Then we heated it with our incubation buffer for 2 hours at 95 degrees. After that, the samples were spun down and the incubation solution was decanted into a 96-well plate with 25 microliters of formic acid. The formic acid was added because after that, we evaporated it to near dryness to make it appropriate so we wouldn't have breakthrough in our sample prep. But we needed to add the acid to make sure we wouldn't lose any of our amine stimulants to evaporation because they can be volatile under alkaline conditions. And then after that, we diluted it with 1.5 ml as 4% phosphoric acid. So now our samples have been extracted, spun down, and then prepared and are ready to be extracted with the solid phase extraction plates. As I mentioned before, this is a technique we've used in the past for other similar panels. We loaded the acidified sample onto the extraction plate. This was a 30 milligram plate. It was washed with 2 washes of 80% water and 20% methanol, and then simply eluted with 2 aliquots of 50-50 acetonitrile and methanol containing 5% strong ammonia. After that, we simply diluted it with a 1/2 ml of mostly water with some acetonitrile and formic acid. That avoided the necessity of evaporating it once more and reconstituting it, and we still had the sensitivity we needed for this assay. So again, the sample prep, we load the acidified samples onto the MCX plate, 2 mils with 80% water and 20 methanol, and then elute with acetonitrile, methanol, and 5% strong ammonia, and simply dilute it and inject it. Our UPLC MSMS analysis was done on the Waters UPLC I-Class Plus with the flow-through needle. Column was a Acuity Premier PEH-C18 column, 2.1 by 100 millimeters with a sub-2 micron particle. Modal phases were straightforward, 1.1% formic acid and water and acetonitrile, and we had a 2 microliter injection volume. And to the right below, you can see the UPLC gradient. It starts at 2% acetonitrile, goes up to 67% before a washout at 90%, and then we're all back to baseline by 4 minutes. In this case, the total cycle time, I believe, was 5 minutes, injection to injection. The MS conditions, we use the Waters Xevo TQ Absolute tandem mass spectrometer, and these are the global conditions we use: ESI positive, capillary voltage, temperatures, and gas flows you see here. And now we are time to our second question, and I'll give you guys the time to take a look and answer that. And this should be interesting because this is what's your biggest challenge when developing a hair analysis method? All right, so it seems like we have people running into issues all over the board, which is not terribly surprising considering it is a tough matrix. So hopefully we can answer some of these questions. Moving on. All right, now we'll talk about the results, including the chromatography. We'll discuss in detail sort of the recovery of matrix effects and how we developed the incubation and the sample prep methods. And then we'll move on to the quantitative results, accuracy and precision, intraday and intraday, and the use of the MSQuant software to analyze this. We also were able to do a comparison with a competitive, not an application, but a competitive extraction matrix or extraction solution. So here are the chromatography results. As you can see, everything is eluting by about a little more than 3 minutes. We have, as I mentioned, we have the opioids, the opiates, the amine stimulants, benzodiazepines, PCP, cocaine, methylphenidate. We also have some nitazines, some of the newer synthetic opiates as well. So a broad spectrum of drugs and metabolites. This is a display of the chromatogram and the Quan Review software. Here we're showing results for cocaine and EDDP. This is probably a calibrator. We see our quantitative ion, our qualitative ion below that, chromatogram for the IS, as well as some data below, including retention time, et cetera. So it's a nice piece of software, makes it very easy and intuitive to review the data. Now let's talk about some of the extraction results. So the recovery, the matrix effect, as well as critical method attributes that we found. So here's our recovery and matrix effects. As I mentioned, this was using our optimized incubation buffer and really represented a compromise for all analytes. As you can see, some of them, such as the amine stimulants, are fairly low. However, we do have plenty of accuracy and the sensitivity needed, so that ended up not being a problem. We were consistent. Let's see. So we also compared this to a commercial reagent. It did okay, but you can see we were, our recovery was average about 60% versus 40%, especially the benzos. We found that those were things like oxazepam and lorazepam were pretty challenging to get out of there. So we're pretty happy with the optimization of the extraction solvent. What are some of the keys to the success? Well, I already talked about the optimization of the solvent, especially for oxazepam and lorazepam, as well as increasing the others. Using the formic acid for the evaporation helps prevent the acid-catalyzed degradation of alprazolam and triazolam, seen when we used hydrochloric acid. To the right, we see some other experiments where we're looking at the recovery of these benzodiazepines. We did try the digestion of the samples, but recoveries were significantly worse and matrix effects were very, very large, often greater than 80% with substantial interference. It was not clean enough, and it also took a long, long time. Many of the digestion techniques I found worked better with SDS, but of course that was almost impossible to get rid of, so we had to do it without it, requiring overnight digestion. Even with that, we were getting some pretty tough samples. And we see in the picture here that brown, it's not a goo, it is a liquid, but it was not the cleanest thing in the world. Now let's take a look at the quantitative results, including calibration curves and our validation results as well. Here are just a couple examples of calibration curves and QC results. These are looking at, again, in MS1, and this just shows the display where we see our curve, and on the left for codeine, we had, I think, highlighted one of the samples. We clicked on it, so it's highlighted in that square box. And then the quality controls, look, you can, it's also, in addition to displaying the curve, it's displaying the residuals as well as the control limits for each compound. So a very easy and convenient way to review the data. And here's just a couple more showing these curves and the acceptance criteria for fentanyl as well as nordiazepine. Here is a calibration summary for all of the compounds from the, again, opiates, the amines, the benzos, the other ones. All of them had excellent R-squared values. The two that we did run into problems with were phentermine, highlighted in yellow, as well as metadesnitazine. Phentermine was subjected to some internal interferences that really limited our sensitivity and ability to quantitate it well. And the metadesnitazine, we didn't have a deuterated or stable labeled internal standard for it. And so we did run into problems with the accuracy of that one compared to some of the other compounds. Other than that, we had very consistent curves for all of the other analytes. As you see, very, very consistent QC results as well. One thing that we did notice with the hair samples is not surprisingly, they tended to be much more affected by matrix effects. For example, meaning that if we did not have a deuterated internal standard, we saw much more variability than I would typically see in a plasma or a urine sample. So that was the one takeaway is, the more deuterated or labeled internal standards you have, probably the easier that your analysis will be. These are within batch statistics. Once again, you see phentermine's not behaving terribly well, but the other ones look very, very nice. Oh yeah. So both, most of our accuracies are within 10%, and most of our CEVs are under 10%. Between batch statistics also look very good. As I mentioned before, metadesnitazine and phentermine gave us some issues. Everything else looked very nice. Again, met all the the typical bioanalysis, forensic, regulatory requirements for accuracy and precision, and exceeded them in most cases. Once again, we see a lot of CVs under 10%, as well as good accuracies. We were also able to run some positive quality control samples. These were sourced from Co-Medical, company in Italy. These were bulk hair spiked with analytes and mixed extensively. They were not authentic samples with incorporated drugs, but they were commercially available. And they gave us nominal values accompanied by reference ranges. So these are the results from those samples. All of the analytes were easily positively identified. A couple, fentanyl and 6-acetylmorphine, were beyond the calibration range. We were looking at lower concentrations than they had. Of the others, 18 of 22 were within the established range of acceptable values. So we're very, very pleased with this result. Again, we were able to see everything and very good accuracy as well. So how do we do? We're able to measure from a single hair sample. We have the sensitivity to easily meet most of the requirements of Society of Hair Testing and the European WDTS. fairly high throughput. We have a short incubation time. It's only a couple hours. The sample prep takes maybe another half an hour, or solid phase extraction, and a 4-minute analysis time. We think the sample prep is fairly simple and certainly reliable. It's straightforward. It's fairly rugged, but we're dealing with a solid complex matrix and multiple analyte classes, so we're able to optimize it for everything that's in our mix. And then our instrument system again had plenty of sensitivity to not only see everything but analyze it fairly quickly. So let's talk a little bit of the advantages. We have a workflow of about 3 to 4 hours versus overnight. The 96-well plate definitely streamlines the SPE process. You could do this with cartridges, but it definitely would take longer. For our results, we have very well characterized extraction, recovery, and matrix effects. One of my high horses I like to get on is a thorough evaluation of recovery matrix effects while doing method development, because otherwise it's very difficult to troubleshoot later on if you start running into inaccuracies or imprecision or something else. So we spent a lot of time really optimizing that, and we feel like this is again a well-characterized method. Again, accurate quantitation for all the compounds except for our couple of troublemakers here. Cost, yes, SPE can increase the cost per sample. My result is worth the trade-off for the results. It's a dirty matrix, and you definitely want to clean it up. And it's often, I find, easier to use than liquid extraction. And so I want to sort of wrap up with a few caveats or considerations. Here's a dirty matrix. We see a lot more endogenous chromatographic interferences than other matrices. We did see substantial matrix effects. They were well controlled with stable label internal standards, but it's definitely more challenging than other matrices. It is a large panel, but certainly could adapt it by adding or removing certain compounds. For example, we did not include ketamine or norketamine, but certain things like that, or any other of the weak bases you could add. And as I mentioned, the workflow has been optimized using a 96-well plate, evaporator, and a positive pressure apparatus. Again, it could be adapted to cartridges, but the workflow would suffer a bit. So, conclusion is a quick, robust, and accurate method for the quantitation of multi-drug panel in hair. The pre-treatment steps have been extensively optimized to accommodate all of the target analytes and MICH. The recommended cutoffs, regulatory cutoffs are met for all the compounds, and the I-class and TQ absolute system has a required sensitivity for this panel and hair, meeting the cutoffs recommended by Society of Hair Testing. And I want to thank you for your attention, and we can take questions now. Here's our contact information. And back to you, Rachel.
Yeah, no, Jonathan, thank you very much for that really great presentation. I think it's really thorough. There's a lot of details that go into this method, especially because of the complexity that you have mentioned. So if you haven't yet, go ahead and please ask any questions if you have any right now. And we will go through those right now as we transition over to the question and answer section. We do have a couple that have come in. One is there's an ask about looking at GHB in hair. Is this something that has been considered or looked at in the past? I know it does provide more challenges even in other matrices, but I'm curious if you have anything to say about that, John.
We have not done GHB in hair. I know, there are a number of publications out there that have done GHB, but it's, I think one of the challenges is fairly polar, and so it's it can be sometimes hard to retain, but you can try it. Again, we have like one of the things is we really optimize the extraction solvent to for a wide variety of polarities. Again from the morphines all the way to the benzos. So I know that GHB is definitely on the more polar side, but I would have to check the pH too, the pKa.
Okay.
And I think it's acidic as well, so that would be another consideration.
Yeah, it could be interesting as a future thing, or if someone is doing it. But good to know. And then another question that came in is, do you do the drug examination in hair as a service for police units, or doing it out of the USA? And I think you touched on it a little bit about the samples that we provided, but
we do not provide any forensic services commercially at all. We're strictly a manufacturer of instruments and developer of example methods.
Yeah. Okay. Wow. We got some questions coming in now, so this is great. One question that did come in, and I think that this is interesting. It could provide. It kind of talks a little bit about environmental toxicology in certain aspects, and it's definitely been an ask. I don't know if we've done much with it, but PFAS is very hot topic when we start to talk about environmental. And now, that there's potentially PFAS in our blood, but haven't really heard of it in hair. Have you thought, or have we thought to potentially try testing for PFAS in hair?
We have PFAS methods for other things, and so I would say that. I think we recently released the specific, solid phase extraction cartridges for PFAS that are specifically designed for it. So I would say it would take some development, but it would be a different, I would say certainly a different SPE extraction, clean up after you're done. I don't know how the solvent system we use would work. We haven't tested it. But,
okay.
That's I'm sure there's other people thinking about it. Carrie might not forgive me if I suggested she do it, but
duly noted. So it's definitely still new, so it's something whether or not it's really been tested. Okay, so Katie has a question. How do you recommend testing extraction efficiency when an authentic positive specimen is not available?
That's a good question. And basically the way we did it was, really develop the best method we could, looking at the literature, making sure that we homogenize it well, looking at our recovery and our matrix effects as best we can. Like you said, without the authentic samples, we can only evaluate as much as we can do. But that's we still plan to get some authentic samples in and run those.
Okay.
Source them from a commercial area, but yeah, it is a challenge, and we dealt with it the best that we could, but as we would do with just about any method development or matrix.
Yeah. Okay. And following up with that, how many isotopically labeled internal standards were used in this particular method?
I think about 50. I believe we had 58 compounds, and we had, I'm not sure, but I think it was around 50. There was a few that we didn't have or were very expensive, like the nitazines.
Yeah. And as
I mentioned, for the
mentioned,
most part, we tried to get them as we could. It definitely made a difference.
And when we publish the application note, this will have this information listed in it.
Yeah.
So it will be included with the application note. Okay, so this is an interesting question. And you probably would saw this maybe more from handling it, but how do you combat static with hair wanting to stick to everything?
It's a challenge. It's definitely a challenge. It depends on the environment. If you're in the desert, you're going to have a hard time. I prefer to do the analysis when it's kind of rainy outside here, but it's, when you're weighing it out, there are anti-static things devices or guns or whatever you can use wing it out, but it is a challenge. And I would say for me, the biggest challenge of that was just, you sort of needed more sample when it's sticking to stuff. It's hard, your 20 milligrams can quickly become only 10 milligrams because it's hard to scrape the staticky powder out of tubes. But it's, again, get a humidifier. It's,
Yeah.
But I found once it's, chopping, for example, when I first snip it up, scissor mince it, make sure to do it within a bag so it's not going all over the place. Things like that. Initially, when you're segmenting it, it's 1 centimeter segment, so it's not too bad. But when it does get powdery, it can be a challenge, especially when it's very dry.
Gotcha. Yeah, that is a
I
challenge.
wish I had a better answer for that.
Yeah. Well, and I know you had mentioned that obviously synthetic cannabinoids were not listed in this particular method. Do we have a procedure for them? Okay. Not at this time.
No, not in hair. I mean, we have other methods for synthetic cannabinoids, and you could, with, a different sample prep. So you could try, again, follow I think our solvent solution that we developed would be pretty useful for them. I think it would work. I haven't tested it out, but it's again, you'd have to look at you'd have to do the experiment. And,
Yeah.
And some people actually no, she was talking about synthetics. Yeah. So
Yeah. And so I'm not sure if we know this exact answer, just because we are doing this more from, developing methods at a manufacturing site. But there is an ask of like, what's the longest time from drug consumption to actually obtaining a hair sample? I think that's more for, those that maybe run these and test them, in production. But you might have.
So hair grows at about 1 centimeter a month. So, if somebody gets a haircut regularly, you only have, a couple months, but it can be stable, there've been published papers of things being stable for years, but I think, for the early incorporation, it would probably, you'd want to wait at least a couple of weeks to make sure it's fully incorporated in the hair.
Mm-hmm. Gotcha. Okay. All right. So,
for how long they get one,
Yeah, right. And then that's very dependable. So one question that came in is, what is the LOD, LOQ you are able to get for the most sympathometric amines, such as MDA, MDMA, phentermine, etc.? Do you know those offhand?
Yes, I do. Those were, let me take a quick look. I believe we were down to, I'll find out in a second.
While he's looking that up, I do want to mention there were a couple of questions asking about the recording of the webinar. Yes, this is recorded. You will get an email tomorrow, most likely, for your thank you for attending, and it will have a link to the recording of this webinar for on-demand use, or if you want to go back and look at any details. So yes, that will be provided to you through the On24 platform.
Okay. Oh, so back to the LLOQ. For most of the amines, we were at 0.01 nanogram per mil. For MDA was the highest one at 0.03 nanogram per mil. And the regulatory cutoffs were 0.2. So we were about 10% or 5% of the regulatory cutoffs.
Nice.
So yeah, 0.01 was most of them.
Gotcha. Okay. And so here is another question. I know we used the information from CoMedical. But were these methods applied to real hair samples that have benzodiazepines, or would you need enzyme hair instead?
Enzyme. Oh, no, they were not real samples. Oh, so the question is, I think, do they need
They
enzyme
need
hydrolysis?
yeah. Oh, hydrolysis. Excuse me. I read that wrong.
Yeah.
Thank you.
Typically in hair samples, or just like saliva samples, you don't need enzyme hydrolysis. Usually when things are, when they're transformed, they're fairly quickly excreted by the kidneys, and they don't really incorporate into the hair very well, from my experience, what I've read.
Okay, great. And so this is another kind of method-specific question. Again, most likely will be in the app note when it's completed. But what was the identification criteria used, such as like relative retention time, MR, et cetera, that was used for how you identified the particular compounds in quantum?
It was the algorithm in the software, but it was, yes, it was, we used relative retention time and, quantitative and qualitative, ion ratios, and the usual, usual criteria.
Gotcha. Okay. Thank you. Just a couple more questions. It looks like here we've still got a few more minutes. And if you have any questions you'd like to ask. So this one's interesting. Has to do with kind of like whether or not we need blank matrices. So how can you be sure that the drugs originated from within the hair and not due to external contamination? What type of blank test needs to be performed with every analysis?
Typically, what people will do is collect their washes when they're washing the hair, and then analyze those washes. And basically, once the washes come clean, they know that they've removed the external contamination. And then, I don't know the frequency with which they do that, but certainly during the method development,
Gotcha.
that is done. But that's typically how you. And
that makes sense. Make sure that
the next one. Yeah.
Oh yeah, and so the next two are kind of more temperature related. Yep. So by heating to 95 degrees during extraction, are you at risk of losing some molecules that you're trying to quantify? It kind of ties into maybe the answer you had for the one previously.
Yeah. That was a concern, but we basically address that by doing a recovery studies. So we would, we had hair samples that had drugs in them, go through extraction, and then we had post-spike samples where we would add the drugs after the extraction, and so we know that we got fairly good recovery for most of them. I think the recovery, I don't think it was due to, to the best of my knowledge, the lower recovery for the means was not due to degradation. The truth were sealed, but I guess that could have been an issue, but it was, I don't think it was. It was fairly, our results were very, very consistent. But yeah, we did the post-spike recovery samples just to make sure. Because if we saw nothing, then we'd say, oh, we have a problem here. But we had good recovery, consistent recovery.
Right. Okay. And then one last question that we have here. Are there any specific temperatures for long-term storage or transportation of the samples?
Typically room temperature in the dark, protect them from UV light, but they typically do not need to be refrigerated or anything like that. You probably want to avoid excessive heat if you can, but it's one of the advantages that, they don't need to be cooled and they're easy to ship and store.
Yeah. Okay. Well, thank you all. That was the last question that we had come in. We really appreciate them. And I hope that this was beneficial for all of you. I know this is a hot topic. We will be coming out with a app note, as I've mentioned, here soon. You have our contact information here if you have any questions for myself or Jonathan. And then be on the lookout as we will be doing some more webinars in the future. We're looking to have one or a couple maybe focused on sports doping and some other topics of interest. So be on the lookout for those. And if you haven't yet, please fill out that survey. We would greatly appreciate that. We really appreciate everyone's attention today, and hope you have a great rest of your week. Thank you.